Going onward, thank you, Susan, for recording. Continuing onward with our conversation and learning and education around this group of pesticides that we're using here in Vermont and learning more about the impact that they're having in our communities and also how we can best manage these pesticides on our farms and around us. So today, we are joined by Samantha Alger from the University of Vermont, and I have had the pleasure of working with Sam for many years now on this topic. And we were just discussing before we went live some new work that we want to do. So it's been great to have a partner like Sam and working on this type of research right here at the University of Vermont. So it'll be exciting to hear what she has to say today. But before I turn it over to her, I want to once again thank the organizations that have provided us funding to do the work that we're doing in this area, both from the EPA, from a pesticide environmental stewardship grant, and also from the USDA crop protection and pest management program. This is an important and critical topic for us to be managing and working on right now. And I'm glad that we have the resources to do that. And as I mentioned on Monday, we have more webinars, more people to hear from as we continue to learn about neonics and managing them in row crops. And on Friday, John Tucker, the row crop entomologist from Penn State, will be joining us. And so be sure to, come back on Friday and listen to John. And then again next week with a variety of speakers, both from Cornell and Ohio State University, talking about IPM and further reducing bee exposure by managing how we plant corn here in Vermont. So I'll be excited to learn more about that as well. We do have CCA continuing education credits, and we will put the QR code up at the end of the webinar today. And we also have pesticide applicator credits. If you need those, you need to email Susan Brulette at uvm.edu, and she'll put that in the chat. Or you can send her your information in the chat if you'd like, and that's easier. All right, so let me go back up here and introduce Sam. Sam is a research assistant professor in the Plant and Soil Science Department here at UVM, and her research aims to improve the health of both managed and wild pollinators through research, education, and outreach. And she conducts research on bee disease, and she teaches introductory beekeeping courses, and she also directs the Vermont Bee Lab. And again, it's been great having a partnership and relationship with Sam. She's taught me a lot, and I'm excited to continue our work with her on this important topic. So Sam, I will turn it over to you.
Thank you so much, Heather. It's been great working with you as well. Really happy that you're putting together this webinar and that I'm a part of it. So thanks I know it's an enormous amount of work pulling these kinds of things together. So really appreciate you and Susan and all of your team's efforts in putting this together, putting this on. Just before I get started here, I would love in the comment section, if those of you who you don't mind, just commenting in there. If you're a crop farmer or if you're a beekeeper, you can just write crop farmer or beekeeper, or maybe, who you are, work for a nonprofit, just a little bit of information about yourself, because I'm just curious about who's joining us today. And I'll get started introducing myself as well. Thanks so much. Okay, so let's see. All right, so Heather gave me an intro, but I'm Samantha Alger. I did my PhD at the University of Vermont. And during that work, I studied disease spillover in managed and wild bees. So looking at how RNA viruses are spilling over from managed honeybees into wild bee populations, which, as it turns out, is a very controversial topic. And so it seems like that's really where my research has been taking me along, very controversial topics, because here we are talking about neonicotinoids. So here we are. And now I'm a research professor here at UVM, and I run the Vermont Bee Lab. So just a few slides about what the Vermont Bee Lab is for any of you maybe beekeepers here in the audience who don't know that this lab exists. We're at Jeffords in room 220. We have a great team of folks who work with us over the summer. Sydney Miller is there on the right, and she's our full-time staff person for the Bee Lab. Shout out to Sydney. She's putting on or leading a webinar on Field Watch, which is on. Sydney, if you didn't mind unmuting yourself and just let folks know when that is.
Sure, thanks, Sam. So yeah, the webinar is on Friday at 1, so right after Heather's webinar on Friday. And FieldWatch is an online mapping tool that allows communication and collaboration between crop producers, pesticide applicators, and beekeepers. So registration is still open for that. Thanks.
Thanks, Sydney. And you can register the same way that you registered for this. There's a link for that, but it is a separate thing. You can stay on after the webinar for the introduction to FieldWatch. And if we get a program like that going on in Vermont, your participation at that webinar is going to be crucial to whether or not we're going to make that happen, because we need to get feedback from you to see if it'll be interesting. So all growers and beekeepers and the public are welcome to join us for that. So there we go. So the main thing that got our Bee Lab started was this diagnostic services. And it's essentially beekeepers can collect samples of their bees, send it into our lab, and we look them over for different pests and pathogens and send beekeepers back a report that helps them with their management practices. And this is a free service for Vermont beekeepers. We also have been managing the Vermont's participation in the National Honeybee Survey, which is a nationwide effort to gather baseline data on bee disease and pathogens. We started this in 2015. The survey also has us collect some samples for pesticide residue testing. So this is some of the pesticide work that we started way back in 2015 or 16. And so results from the National Honeybee Survey can all be viewed online. And we also have a number of research projects. Mostly, it seems like we team up with beekeepers on things that interest them, and then we pursue them, is kind of how our lab has been working. So we have a project where we're supporting bee breeders and incorporating tests for hygienic behavior in bees, and helping bee breeders in the field and in the lab, leveraging our ability to test for pests and pathogens to help them make selections for more disease-resistant bees. And that's been a really awesome opportunity to work more closely with our beekeepers in the community. And then we also have, starting in 2021, we started monitoring for pesticide residues. And this largely came from interest from beekeepers to learn some more about the health of their hives and what kinds of chemicals their bees might be exposed to out in the environment. And we started doing that in 2021, and we've been doing it every summer since, and it's just been growing. And I'll be sharing some of the results of that work during this talk. And then lastly, as Heather mentioned, we teach introductory beekeeping classes, and those are open to the public. It's a 4-week hands-on intensive beekeeping course in the summer. We also have an online version. And hey, check it out if you're interested. Shameless plug. Okay, so that's enough about me and the Bee Lab. So an outline for today's talk, I'm going to talk about the pollinator trends, some of the properties of neonicotinoids that make it noteworthy for talking about bees, the exposure routes for how pollinators are exposed and how they're impacted, some little bit of background information for understanding toxicity, like what is an LD50, what is a part per billion. Those are the kinds of things that keep cropping up, I think, when we talk about neonics, and sometimes it's helpful to just start from square one to understand what those things mean. A very short review about some of the work that's been done looking at differences in crop yields with and without neonics. And then I'm going to introduce some of the pesticide monitoring results that we've been seeing with our work, and then some slides about how folks are working together to reduce the impacts of neonics on pollinators. So a lot to discuss in a short amount of time here. Okay, here we go. So insects are important for our agro ecosystems because, of course, they're decomposers. They provide pest control. They're really the base of our food webs, and of course, they're pollination services. Through the pollination of pollinators, they provide $235 to $577 billion to the agricultural economy. But they're also important for the pollination of our native plants, of course. And oftentimes when we think about bees, our mind goes to this pollinator. It's the European honeybee or the Western honeybee. It is an incredible species of bee. They're the iconic species of bee, but they are an introduced non-native species. And they're only one of our 350 species we have here in Vermont. They were brought here by colonists because they produce honey and wax, so things that we can enjoy. And now they provide much of the pollination services of our food crops because of the way in which we grow food in these large monocultures, and the need for these incredible pollination service events that occur in a very short period of time. And honeybees are great for that because they can be managed in these little boxes and placed down into crops and moved around on the flat beds of trucks to different crops throughout the year. And so because they are actually a managed livestock animal, much like cows or chickens or pigs. They're a managed agricultural livestock animal, which is incredible, right? An insect that we farm. And because of that, because we have beekeepers who are opening up their hives and assessing the health of their honeybees all the time, we have a much better handle on the health and loss of honeybees than we do on a lot of our native bee species, because native bees are harder to track for obvious reasons. A lot of them nest underground, for example. And so, I'm going to present some of the information that we have about colony losses with honeybees. And I oftentimes think of honeybees as being sort of this indicator or canary in the coal mine or indicator of the broader insects issues that we might be seeing. Because they're so closely managed. So this is some results from a survey of colony losses that's put out every year by Beamform Partnership. And it's there's lots of different colony loss surveys that are put out, but this is the best one for a wide geographic area, and they've been doing it for a long time. And so since 2010, Vermont beekeepers report losing somewhere between 15 to 85% of their colonies each year. And if we look at last year, Vermont beekeepers reported losing 42.3, so 42% of their colonies in last year. And so beekeepers feel that these numbers are not sustainable to their operations, losing half of their hives every year. But for the good news is, at least for us who like to eat the food that's pollinated by honeybees, beekeepers are really, really good at making more bees. They can make splits if they have healthy hives coming into spring. They can take one hive if it's really strong and healthy and make it into 2 or even 3 other colonies. So despite the fact that beekeepers might be losing half their hives in a single year, that's the reason why year after year we don't see a disappearance of all honeybees, or we often read these headlines like honeybees might be going extinct. Like, that's not going to happen as long as we have beekeepers who are willing and able to make more bees. However, it's clearly a lot of work. It's very laborious and it's very time consuming. It's very expensive for beekeepers in this industry to keep making more every year. And it wasn't always like this. And so, what's going on with the native bees? There are roughly 20,000 species of bees on the planet. There's 4,000 or so in North America and 350 species just here in Vermont. And thanks to the great work from the Vermont Center for Eco Studies, just last year they put out this report, The State of Vermont's Wild Bees in 2022. And this really, I think, put Vermont ahead in our understanding of what our native bees, the status of our native bees. And I would argue we probably, thanks to VCE, we probably have a better idea of our native bee conservation efforts that are needed than anywhere else in the US. And so we know through their work that over 30% of Vermont's native bee species are ranked as critically imperiled or imperiled, and 55 of Vermont's 350 species are in urgent need of conservation action. So we know that beekeepers are losing honeybees at an unsustainable rate every year. We know that the native bees are also many of the species are experiencing declines, and we're seeing losses there. So what's causing all of these issues? And of course, we know it's not one thing, it's a multitude of interacting stressors like, let's see, can you guys see my cursor? Yeah. Pests and pathogens, of course, climate change, the loss of non-contaminated flowering resources and habitat. That's an important thing. Of course, they need habitat for nesting and they need flowers to eat, and exposure to pesticides. And because I'm here giving a talk about neonics, we're going to really be focusing on insecticides and in particular neonics. So since I'm one of the first talks of this series, I thought it would be helpful giving a little introduction around neonics. They're a class of systemic insecticide. They're used as seed coatings in the majority of row crops, so corn, soy, and wheat. We saw yesterday a talk, or not yet, I guess 2 days ago, a talk from the Agency of Ag, and in their reports, over 99% of the corn grown in Vermont are grown from treated seeds. I think it was 35% of the soy are grown from treated seeds. Other uses of neonics include foliar sprays, soil drenches, and trunk injections. And for crops, we see neonics being used on fruits and vegetables. And one of the biggest use of neonics, besides the use as treated seeds, is actually on turf grass. So think of our golfing and ornamentals as well. So I borrowed this slide from Xerces, and it outlines when we think about, from the perspective of a pollinator, of a bee or a butterfly, what makes a pesticide most risky for pollinators? And these are some of the biggest things: how toxic it is, so whether it has a high toxicity, how much it's used, and whether that pesticide moves off site easily, persistence, so whether it has a slow degradation rate, and then the variation of exposure pathways for pollinators. And we can see, I'm going to go through a little bit of the properties of neonics, and we can see that neonics sort of check every box here, making that group of pesticides particularly risky for pollinators. And one of the reasons why I think we're in the process of talking about it today is because of some of these reasons. So neonics are highly toxic to many classes of insect, and they were introduced with great fanfare because they're considered to have reduced vertebrate toxicity when compared to other insecticide classes currently in use. They are systemic, and because they're relatively small molecules that are highly water soluble, the neonics are taken up by the plant, and their compounds are circulated throughout the plant tissue, which provides a period of protection against a number of sap-feeding arthropods. So this systemic action is a key characteristic that makes neonics so great, and for their use because of their flexibility in methods of application. But we know that because of systemic pesticide that's taken up into the plant tissue, they're exposed also in the nectar and pollen of those plants. And as we'll learn a little bit later, that's one of the reasons why they're very problematic for pollinators that are collecting this nectar and pollen. So neonics are very persistent in the soil. I looked up and found these ranges here, and I myself was pretty surprised at this 20-year for clothianidin, but it is cited, and you can look at these citations down here. So the half-life, so this is the amount of time that it takes for half of the amount to degrade. For these are some of the three most common and most toxic to pollinators. So clothianidin, imidacloprid, and thiamethoxam. You'll hear those later on in the talk. Clothianidin can persist from 148 days to 20 years. It depends on the soil type and UV degradation, of course. Imidacloprid as long as 3.5 years, and thiamethoxam just about a year. So degradation is obviously higher at soil surfaces due to UV degradation. And my next slide sort of demonstrates that. Now, I hope Heather doesn't mind, but I took some of her results that she presented to the Ag Innovation Board because I thought this was really the best example of some of the persistence and maybe movement of neonics out in the environment that I've seen. And these data were collected here in Alberg in Vermont. And so these were samples that Heather and her team collected in fields that had no historic use of neonicotinoid treatments. And at the very surface of the soil at 0 to 2.5 inches, there was no clothianidin detected, probably or maybe likely due to UV degradation, or the fact that this field had never been planted in neonics. That's another potential reason. However, if you look just under the surface of the soil at 2.5 to 6 inches, 75% of those samples, her team was able to detect clothianidin and at a average concentration of 6 parts per billion. And if that number doesn't mean too much to you right now, it will make more sense as we get on and talk about toxicity. But that was pretty surprising to me, and indicates, in a field that had never been planted in neonics, it indicates how these pesticides can move across the landscape, either under the soil or through dust into non-target areas. And it's really just, in this case, just below the surface. So generally speaking, the way that pollinators are exposed to pesticides are, direct, where they get sprayed directly, which is probably the worst case scenario. Where they visit a flower that had been sprayed maybe the day before or sometime previously, so contact residue that's left over on a flower. Lots of solitary bees will collect plant tissue and soil to line the inside of their nests. And so if that plant tissue or soil has been contaminated, they're bringing back those materials back to their nesting site. So that's another way that they can become exposed. And then, of course, contaminated nesting areas. So 75% of our native bees are ground nesting bees. And so anything that's sprayed on the ground or might or persist in the soil is a way in which pollinators may become exposed to these to pesticides, just broadly speaking in general. But a pesticide that takes a long time to degrade, clearly that a bee will have lots of opportunities to come in contact with that if they try nesting in the soil. So with neonics, there's some other interesting exposure routes that it can look a little different, particularly when you're talking about neonicotinoid treated seeds. There are other things to consider. So with neonic treated seeds, the seeds are planted in the ground, and the seeds and as the plant grows, the plant takes up somewhere between 2 to 3% of that chemical into its leaf tissue. And the other 90% or so remaining insecticide on that seed coating leaches out into the soil, where it can then end up in waterways where aquatic invertebrates can become exposed. And then it can end up contaminating soil. And neighboring plants that are also growing in those contaminated soil can take up those neonics into their plant tissue. And we've seen it in lots of previous studies and a little bit in my work as well. Another thing to note is as these seeds are getting planted, actually I've got a slide on this. I'll just go to the next slide as we talk. But I think, well, yeah, as the seeds are getting planted, there's a dust that's created. So these seeds are coated in a talc or graphite powder typically. And that graphite or talc powder is laden with neonicotinoid residues from the seeds. And the wind will blow that dust across the landscape and could end up in non-target areas. And I thought it was interesting to note that the same amount that's taken up by the plants, 2 to 3%, is the same amount that's actually lost to dust. So this dust obviously is another exposure route for pollinators, and concerns around the dust actually started in 1999. I did a little bit of research on this because I was wondering when was the first time we started being concerned about this. So concerns regarding this pesticide contaminated dust, actually, it followed atypical honeybee losses in a number of countries and it was reported in Italy, France, Slovenia, Germany, US, and Canada as far back as 1999. And then a paper came out in 2003 that really implicated the dust as a potential route to honeybee exposure to neonics. So this was 20 years ago that we've been, that we've known about this and have been learning about it and trying to figure out what to do. And then in 2017, this was a really interesting study that set up dust traps around a field and measured how much of the residues are leaving the target area into non-target areas. And so this is, I know this might be hard to see, but this is downwind and this is upwind. And the blue represents the margin of the field. And this heat map shows the, I guess, level of residues in the dust and where it ended up. And so you can see a lot of it is leaving that field into these margins here. A study in 2012 found that clothianidin is detected up to 9 nanograms per gram in dandelions. That number is 9 nanograms per gram. You'll be hearing about parts per billion and these kinds of things a bunch. But just to kind of put that in perspective, that's a level that it will not induce acute toxicity in honeybees. So if a bee were to eat that or get orally inoculated, or it were to touch its body, it wouldn't just die, but it's definitely high enough to cause sublethal impacts, which we'll talk about later. But in let's see, 2012, it was found that the dust clouds that are around the planting machines reach concentrations that are sufficient enough to kill bees just passing through in a single flight. And this really interesting work where researchers conditioned, so they actually taught honeybees to fly through the generated dust clouds and showed that they acquire lethal doses of neonic residues in flight with concentrations ranging from 50 to 1200 nanograms per bee. And to put that in perspective, that's 60 times the lethal dose that bees, 60 times the lethal dose of 20 nanograms per bee. So there's, I guess I have a little video here of let's see if I can play this. Okay, yeah, of someone planting, and you can see a little bit of dust coming up from that kind of that imagery. So there's a coincidence here of seed sowing and the bloom of spring flowers like dandelions that may create scenarios where the flight path of bees are really very likely to overlap both in time and in space around some of these fields that are getting planted. And as a result, bees are at greater risk of in-flight exposure around these agricultural areas because of this planting time coincides with these really important nectar sources like dandelions that are blooming around these fields at the same time these things are getting planted. All right, see if I can. So because neonics are systemic pesticides, the chemicals are taken up into the plant tissue as it grows, offering it protection from pests, but the neonics are also expressed in the nectar of pollen. And so while corn doesn't produce nectar, it does produce pollen. And I think you can see we captured this footage last summer of honeybees collecting pollen from corn. And so I think corn is typically considered a wind pollinated plant, but surprise, like honeybees will visit corn. And I often hear that, we're not concerned about honeybees visiting corn because it's wind pollinated, but in fact they will. And previous research has found that the residues can range between 1 to 6 nanograms per gram in corn. And to put that in perspective, we know that at levels of 5 nanograms of clothianidin, new queens are less likely to survive, less likely to lay eggs. Eggs that are laid are less viable. And at ranges of, let's see, well, we'll just stop there, but you can see that these tiny amounts actually cause these sublethal impacts, which unfortunately are very hard to measure, which we'll talk about in a minute. So since most of the neonics are leached into the soil, like 90% from those seed coatings actually don't end up in the plant, but in the soil, neighboring plants can take them up from contaminated soil, much like how nutrients are taken up into a plant. And so, this is another way in which, dandelions and some of these other flowering plants around cornfields might take up neonics into their leaf tissue, but also in the nectar and pollen of the plants. So understanding impacts of neonics to pollinators, I think oftentimes when we think about a pesticide exposure, we have a vision of something like this, which shows piles of dead bees outside of a hive that would signal to a beekeeper that there was a pesticide exposure event. Or to an apiary inspector that there was an apiary inspector might get a call if something like this happens. And that's a clear exposure event. And while this can definitely happen with those acute exposure events, like I talked about earlier, sublethal and chronic exposures don't look like this, and they are much more difficult to measure. And we know from the extensive research that's been done that sublethal exposures can make bees less able to forage, grow larvae, and fight off disease. And so these are all things that over time would cause a reduction of the workforce in the hive, and then maybe eventually death. Maybe their immune system might not be able to ward off a virus. But it's not gonna look like this all the time because of these sublethal impacts. But when we look to field studies that have been done looking at realistic levels of neonicotinoids, we find a reduction in wild bee diversity in sites that are field sites that are planted with neonicotinoid treated seeds compared to fields that are treated with untreated seed. Solitary bee nesting is also reduced. Bumblebee growth rate and reproduction is reduced. We also see studies that found a reduction of overwintering success, colony reproduction for both honeybees and wild bees, and decreased survival and immune response in honeybees. So that's where I was getting at, maybe they're less likely to fight off a virus is because their immune system is also impacted, and that's another sublethal impact of these neonicotinoids. All right, so now we're going to be getting into some of these, how to measure risk and hazard to pollinators and kind of breaking down what parts per billion and LD50 and all those things mean. So when we measure risk or measure hazard to beekeepers, this takes into account both the exposure levels. So that means the level of the pesticide residue in the sample, like the pollen, nectar, or wax, the level that you're finding out in the environment, and then the toxicity of that pesticide, which is typically measured as an LD50. So we've got 3 different neonics listed here: clothianidin, imidacloprid, and thiamethoxam. And LD50 could be measured as either the contact LD50, so it means that it's touched to their body, or oral means they've ingested it. And an LD50 is the amount of a pesticide which kills 50% of the honeybees in a test population. And so the smaller the LD50, the more toxic the pesticide is, because it meant a smaller amount was needed to kill half of the pollinator, or half of the test population, half of the honeybees in that test population. And so these are the LD50 values, the contact LD50 values for these 3 neonicotinoids. And I have this preference towards leaning towards presenting everything in parts per billion. So that's what these values are expressed in, the units of parts per billion. And just as a little conversion, 1 part per billion is the same thing as 1 nanogram per gram. And so what is a part per billion? Heather had actually recommended that be something that I talked about, and I thought it was a great idea. So to kind of wrap your head, like, what is 1 part per billion? It's the equivalent of 1 drop of impurity in 500 barrels of water, or 1 cent out of $10 million. And so you can imagine it's a very, very small amount, right? And if we look at the oral LD50s for these neonicotinoids, we can see that that's a lower amount. As you would expect, a chemical would be more harmful to you if you were to eat it than it were to touch your body, right? So that's why we see these much lower levels. Much less is needed to kill half of the honeybees in a given test population. But then we also have this LOEC value, and this is where it gets into, I think, in my opinion, and a lot of other opinions of scientists, it becomes more biologically relevant. So the LOEC is the lowest observed concentration that produces an adverse effect. So let me just go back here, and you can see that those numbers are much, much lower than those contact LD50s and oral LD50s. So LD50s, they underestimate the impacts to animals because they only take into account death, not all of the other problems that might happen up until death, the physiological, behavioral, reproductive impacts that occur actually before death. So LD50s are certainly important, but I think that's one of the reasons why we should be looking at LLEC values to determine whether when we're testing, sorry, take a step back. When we're testing for pesticide residues out in the field, it's important to use labs that have equipment that are sensitive enough to detect these tiny levels, because it's much more biologically relevant than looking at these LD50s, because at that point we have half of a population that's dead. All right, so this is a really interesting slide that I borrowed again from Xerces. And let me see here. So over the past couple decades, overall the volume of insecticides applied to crops has declined. And so instead of talking about pounds per acre as you would with pyrethroids, we talk about much smaller amounts, like ounces per acre with neonics, because we need much less of an amount. But what this chart is showing from this study here found that the total toxic load to honeybees in the US from insecticides has increased over time, even though the overall use by volume has gone down. And so that's what this y-axis is showing. It's an index of honeybee toxicity, and it's calculated from the total amount of insecticides used, their acute toxicity, so their acute contact toxicity to honeybees, and their environmental persistence. And so we can see that it's gone up, but it's largely, it's largely because of the increase in use of neonicotinoids that we've seen such an increase over time. And when we break this down by crop to understand, like, well, which crops are really contributing to this increase in toxicity, we see that corn and soy really make up the majority of these of this. And this is largely because of the use of neonicotinoid seed treatments that are used in these crops. And there are over 36 million hectares of treated corn, 16 million hectares of treated soybean fields in the US as of 2018. Okay, so now I'm going to talk a little bit about crop yields. I thought I had another slide up here, but I didn't. So the thought is that, we're using these neonicotinoids in about 99% of corn in the state, according to the recent survey and the great work that was done by the Ag Innovation Board to get a better handle on how much of our treated seed is being treated with neonicotinoids. So we're using it in over 99% of our corn here in the state. And so the question is it effective? Are we seeing differences in crop yields as a result of using this product? And Cornell in 2020 did this study where they reviewed hundreds of field trials that compared the yields of corn between, and other things, but this is a field this is a figure just showing corn. Compared the yields of corn between fields planted with treated crops and non-treated crops and alternatives. And what they found was that the vast majority of instances, treated seeds did not benefit crop yields. And the story was similar with soy actually too. And so I'll just explain what this pie chart means. So this is giving you the effect of neonicotinoid treated corn seeds on yield compared to untreated. So these are seeds that aren't treated with anything. And then this is showing you seeds that are, it says non-insecticidal seed treatments. These are seeds that are treated with just fungicides. And so when you compare treated seeds with non-treated seeds, we see that out of, the over 300 trials, there was a 13% of the time there were positive effects, but the vast majority of the time, all this gray area, there was no difference in using these treated seeds compared to untreated. And then with fungicides, you see something very similar in that 11% of the time we're seeing you see a benefit to using treated seeds. So the take home here is not that there's not a benefit all the time. The take home here is that statistically there's a small chance of a benefit for growers. And so the question is, does it make sense for nearly 100% of farmers to be using these insecticides when there's only a small chance of it actually bringing a benefit to the crop? And I do understand the importance of, you know, maybe using these seeds as a cheap insurance policy, which is, I think, what the Ag Innovation Board was saying the other day, is that these seeds are actually being used as a cheap insurance policy in the case that when something does go wrong, they're protected. But when you take into account all of the impacts that these insecticides have on the broader environment, and you know, it's a question of, is the 99, does everyone need to use the seeds? Perhaps not. And it's, you know, it's important to note when farmers should have a choice of whether or not they need to buy it or not, which seems to be an issue because it seems like all seed is arriving with seed treatments, and farmers have very little choice about what they can buy right now. So this is another slide that I borrowed from the work that Heather Darby and her group is doing this great research. And she presented this work to the Ag Innovation Board. And so this is another example of what I just described here. She grew, her crew grew, did similar trials where they had treated and non-treated crops of corn, and they compared their yields and found something very similar, that there were no significant differences seen across treated and untreated seeds in terms of corn yield. So it's also here in Vermont. This is a 2-year study, and this was just year 1. So I'm very interested to see what happens in the second year. But this was another example of a trial that's happened here in our state. So now I'm going to talk a little bit about this question. So are Vermont bees exposed to neonics? This question has come up multiple times, like, well, let's see what the data show on pesticide exposures to Vermont bees. And for a long time, there was very, very little data on pesticide residues in hives. And so I had an undergraduate student who was interested in starting to do this work. And so in 2021, she set up these pollen traps on hives on about 16 sites. Let me see, no, 4 sites throughout Vermont, and sample them monthly. And a pollen trap is it's either a series of screens or these little like small openings. And so as the bee goes into the hive, the pollen that's collected on their legs will get pulled off or scraped off, and it will fall into a trough. And so it's a really amazing way to use the workforce of the honeybee to sample a wide geographic area, because honeybees will travel 2, 3, 5 miles from their hive. You can get a pretty good idea on average, like what is out there in the environment that bees are foraging on by setting up these pollen traps. And you can turn them on and turn them off. And so we only turn these pollen traps on in the on position for about 2 to 3 days at a time. So that's important to note, because the results that I'm sharing you is kind of like a snapshot of what the bees were bringing back into their hives in a 2 or 3 day period. And these little pollen loads that you see here are different colors because bees are foraging on different plants during their foraging bouts, and different plants have different colored pollen. And so you see it'd be very colorful. And so all the samples that we've sent out for processing after we collect these pollen samples are sent to Cornell, to their chemical ecology core facility. The cost of the sample processing is around $90 a sample. So it's not cheap, but in terms of analyzing samples for pesticide, this is a really good price. And the services are available to beekeepers, farmers, government agencies, researchers, pretty much anyone who wants to send in a sample. So I'm just throwing it out there if you guys want to check out the lab. They do currently a 93 pesticide multi-residue screen. So other labs might do more pesticides, but the reason why we use Cornell is because their limits of detection are less than 1 part per billion for most pesticides. And that sort of touches on what I was talking about earlier, that if you want to be analyzing samples for pesticide residues with an eye towards their impacts to pollinators, you really have to be using equipment that can detect pesticides at a level low enough that we would be able to capture the levels that are known to impact pollinators at those tiny, tiny amounts. If you use a lab that has a limit of quantification of like 10 part per billion, you're going to find levels. So much of your samples are going to come back as false negatives. So this is, here's a great picture of the pollen load on a honeybee, if you weren't quite sure what that means. But yeah, so that's what they're coming back to their hives with, and that's sort of what gets scraped off into their pollen trap. And each pollen load consists of lots and lots and lots of pollen grains, and they collect them from flowers and use their saliva to stick it together in a ball and stick it to their legs. And they've got these concave areas on their legs that are able to capture these pollen loads on their bodies. So a little bit of bee biology there for you. So I thought it'd be interesting to show how far a pollinator might fly throughout a landscape. And so these 3 circles here represent the flight range of 3 different apiaries. And so the apiary, these are 3 different apiaries in Addison County, and there's an apiary located at the centroid, or the center of each of these circles. And this is the average or median, I'm sorry, the average flight zone of a honeybee, and they might fly for about 2 miles. So this is a 2-mile radius, and so the bee can potentially come in contact with any flowers within this given area. And our lab was interested in seeing how much of the corn and soy might be found within each of their flight zones. How often are they coming in contact with potentially neonicotinoid treated crops? And so the red, we used some land use cover data to just classify the corn and soy. And so the corn is red and the soy is purple. And so you can see for these 3 particular apiaries, there's a lot of these crops within their flight zones that they may be coming in contact with. So in our first year of sampling, we had a really great design where we looked at 4 different apiaries and tested them each month. And so I'm showing you the way that those pesticide detections were classified during that time period. And so we're not looking at just neonics in this instance, we're looking at all of these different fungicides, herbicides, insecticides, miticides, synergists as well. And so this is how they're broken out into those different categories over time. And this is the percentage of the sample that was of a particular sample that we found fungicides in and herbicides and so on and so forth. And so, these are the months, May, June, July, August, September. We can see that if we're interested in looking at neonics, it's the yellow bar. And we can see that most of the neonics that we're finding in our honeybee hives during this project were found in May, some in June, and some in August. And so we hypothesized that this uptick in neonics in the early part of the season might be due to the dusting that's happening during planting time, during the corn and soy planting, when the dust, laden with neonics travels across the landscape and lands on things like dandelions and apple blossoms and all of that. So we predicted that, and then we were thinking that maybe the other reason why that they're cropping up again in August might have something to do with when corn and soy are blooming. And so I don't have a definitive answer for you yet, but we've since changed up our sampling protocols to focus on these time periods to see if we can find more neonicotinoids during dust planting time and when corn and soy is blooming, to see if we can dig into that a little bit further. But our overall results over the past couple of years, so in 2021, we had only 16 samples, but $90 a sample when you're trying to get money together to test these things. We had 81 detections of 20 different pesticides in 16 samples. And that to us was pretty surprising. You think that these sampling efforts are really only a day or 2 at a time. So samples range from having 2 to 13 pesticides in a single sample and an average of 5 pesticides per sample. So that's not just neonics, that could be fungicides as well, or miticides. It could be lots of different things. But it was still a lot to think of that these bees are coming in contact with up to 13 different chemicals in a single, in a 2-day period as they're coming back into their hives. In 2022, we looked at just a few more samples and found 89 detections of 23 pesticides. And then in 2023, we got some funding for this work, so that was great. And so now we collected 100 plus samples, and we don't have results for that work yet, but we should probably within the next month be able to share the results from 2023. But when we look at just neonicotinoids, for this work, we found that about a quarter, or if we're looking at the three most considered to be highly toxic by EPA standards, clothianidin, imidacloprid, thiamethoxam, 21% of the pollen samples were positive for at least one of those highly toxic neonics. And whenever we found neonics in the pollen samples, they were always found at a level above that LOEC value, so above the level that's known to cause those physiological or reproductive or behavioral impacts to pollinators. But they were not above the LD50 values. And we did find, we had a very small sample size of plant tissue. We started just collecting plant tissue last year. We only had about 7 plants that we collected, but we did find clothianidin in goldenrod flowers blooming adjacent to some cornfields. So indicative that they are taking up these chemicals from the soil and expressing them into their leaf tissue and flower tissue. As you probably all know, goldenrod blooms later on in the season. And so it was probably not, it was not like a dust event where the dust landed on the flower, because they bloom, you know, in August and September. And we know that these corn and soy are being planted in much earlier in the summer. And so this is probably indicative that that plant is taking it up from the soil. And so, you know, it really, as I'm driving around the Vermont landscape and looking at all these beautiful goldenrod and fall asters that are blooming around August and September, I'm starting to think about this happening, and it's been an interesting perspective to have after finding neonics in these a really important forage plant for both honeybees and native pollinators. And honeybees rely on goldenrod to make their fall nectar crop, and that's what they eat throughout the winter. So ways that efforts that are being done to reduce impacts to pollinators. So there's been lots of mitigations that Heather and her crew are working on, which is great, including reducing this dust. We've known for a long time, 20 years plus, that this dust is an issue. And so looking at how these fluency agents and planter modifications can happen. I'm not an expert on this, but you are getting a talk on it later on, I think by Reed Johnson, talking about some of these ways that we can reduce impact. There's also been discussion that was brought up by the Ag Innovation Board around reducing pollinator habitat around crops, which I would not, it does not align with my thinking, is that reducing the pollinator habitat might not be the way to improve pollinator health. But I do understand the thought of trying to reduce the exposure around the crops. In Quebec, in 2019, there were some regulations around a justification of need program through pest scouting. And I'm really interested to learn more about that. And I'm working on pulling together a panel of Quebec farmers and agronomists to learn more about how this has been working for them. You know, there's been, because of this justification of need, it's almost turned into a ban of neonics in that area, and they're just our northern neighbors. So it'd be great to learn more about how they've been weathering this, and whether they've seen differences in crop yields. And so I'll talk about it in a little bit, but I'm thinking about pulling together a panel of Quebec farmers, and Vermont farmers would be invited to have this discussion and learn more from each other. And then in New York, there was a bill proposed to phase out neonics altogether. And I thought it'd be interesting to mention it. It's this birds and bees bill, interestingly named. It was passed by the New York Senate and Assembly in 2023. And the governor has until December of this year, so maybe about 15 days or so, to veto or sign that bill. And what this bill would do would be to phase out the use of neonicotinoid treated seeds for row crops in corn and wheat and soy, and also a ban on ornamental and turf grass. This is the first bit of legislation that I'm aware of that really directly would phase out or ban neonicotinoid treated seeds in the US. And this is our neighbor state. So it's going to be very interesting to see what happens from that, and whether it's changed or amended or passed or vetoed. It's going to be interesting to see what happens with that. So talking a little bit about Quebec, I had a conversation on the phone or on Zoom a couple days ago with Louis
Hey,
Rob.
Sam, sorry to
Yeah,
interrupt, but there's actually only 4 minutes left.
I've got 2 slides.
Okay, great.
Okay.
Okay, awesome. Keep going.
Okay. Yeah. Um, and this was the experiences that were shared by Louis Robert over the phone with me, and that he has seen a decrease in treated seed use. So it went from 2015 from almost all corn and soybean were treated to 0.5% of corn being treated in 2023. That seed companies reacted to the supply by supplying non-treated seeds, and that also insurance companies reacted accordingly. There has been concern around insurance companies not supporting the purchase and use of non-treated seeds. So I guess insurance and seed companies reacted to the consumer up in Quebec. And that some farmers moved to diamide insecticides, but he said others have abandoned insecticide treatments altogether. So whatever policies or best management practice or anything that gets drafted, I know that farmers are going to have very valid concerns, and this is great that Heather and is putting together this webinar to kind of talk about all these things. And these are just some of the concerns that I'm hearing about from farmers and groups around any policy changes and that might get drafted or best management practices that are adopted. That field trials need to continue to investigate crop yield impacts. We need to improve the availability of untreated seed. There needs to be technical support for pest scouting and sourcing for these fluency agents, guidance on crop insurance and what that means, and then offering opportunities for sharing experiences. And I'm going to try to focus on that one, hopefully during this panel discussion with Quebec farmers. And you all would be invited to join for that if you are available. And yeah, sorry, I did talk for almost the full hour.
No, that's fine. No, it's great. And that's what people are saying. Great presentation. There is one question that came in. And again, if you have questions, please put them in the chat. And I'm going to actually put up the QR code for the CCA credits in case anybody needs to leave. And again, if you need pesticide applicator credits, please email Susan. But somebody's wondering if there have been tests to detect neonics in surface waters.
Yes, thank you for bringing that up. There's a 2022 report from the Agency of Ag looking at neonics in surface waters. And the only county I think that where they detected the neonics is in Franklin County right now. And it was clothianidin at levels, well, I don't remember right now, but is there a way to send information to people after post? Maybe I can put it in the chat if I can find it real quick. But if you search 2022 surface water report from Agency of Ag, you'll find those results.
Great. And Aaron is asking, is neonic use on other crops also a problem for bees?
Yes, of course. And the reason why we're focused, or my research has been focused on these neonicotinoid treated seeds is because of two reasons. It's the biggest use of neonics in the state, and the fact that there's been so many trials that have found that it does not cause a big, for most of the time, it doesn't result in higher crop yields. And whereas it might get a little grayer with some of the vegetable and fruit crops, but it definitely is impactful for pollinators on any crop.
Great. Is there residue in the beeswax and the honey?
Yes, residues stick around for a lot longer in beeswax, but I haven't tested, I have not tested honey.
Great. And there's Morgan, thank you. She's putting some links into the chat
Thanks,
from the Agency of Ag. And then also Emily is mentioning that she is going to be working on testing surface waters in Vermont and New York this coming year. So that's really great.
Yeah, I think one of the links that Morgan dropped in there was the surface water report. So that's
good. Perfect.
Morgan.
Thanks, Morgan. Well, this was really great, Sam. Again, thank you. I mean, really appreciate all the work that you've done and time and effort focused on this topic. And as Sam said on Friday, there's an additional webinar that follows John Tucher's webinar talking. Can you mention it one more time, Sam, so people know? I don't want to mess
Yeah,
up.
is
mess it
Sydney
up.
still on here?
Yep, I see
Yes.
that.
Hi.
Do you want to introduce your webinar again after John Tucker's talk?
Oh, sure. Yeah, so the webinar is about Field Watch. It's from 1 to 2 on Friday after John Tucker's talk. And Field Watch is an online mapping platform that allows communication and collaboration between crop producers, pesticide applicators, and beekeepers. So definitely stay tuned for that. And your participation in that webinar will determine if we seek to fund Field Watch here in Vermont. So definitely
Great.
check it out.
Thanks,
Thanks.
Sydney. And
Yes,
just so people know, it's a separate link. So you're gonna, which was on the flyer, and you'll have to register in a different way. So make sure that you do that. It's on the flyer again. There's a link. So sorry, I don't have it handy to put in the chat right this second. But
I can throw it in the chat.
okay, thanks, Sydney.
Sure.
All right, so I'll let Sydney do that quick, and then we'll hopefully see most of you on Friday. And thanks everyone for participating. And thanks, Sam, for taking time out of your busy day to be here with us. And there we go. Susan just dropped it in the chat. So perfect.
Thank you, Sydney.
All right, everyone, have a great day.
Thank you.
Bye. Thanks, Sam.
Yeah, I'm getting over a cold. I don't know if